Here is to 5.3 Mendelian genetics.
Just as a disclaimer for AP Biology, you are not expected to note the historical information as it relates to Gregor Mendel. We now know Gregor Mendel is a 19th century Australian monk who is considered the father of genetics. However, you don't need to know all of these details for your exam. It just helps to create a picture so you can really relate to pea plants and its understanding as it relates to topics like Punnett squares and genetics. So let's talk about the nitty-gritty. We know that genetics is the study of heredity, which basically focuses in on this idea of how physical attributes are transferred from parent to offspring. Gregor Mendel considered studying pea plants and utilizing his own experience with them through experiments and tracking the traits through multiple generations. What we mean by different traits, we're talking about the color, the texture, the height of the pea plants. This really allowed for multiple traits to be studied. And because pea plants can grow rather quickly in a lifetime, specifically a human lifetime, Gregor Mendel was able to study various offspring over a period of time. And he uncovered these patterns of inheritance and now serves as a cornerstone of what we now consider modern genetics. And that's what you need to understand about that topic. We're going to focus in on what he learned when covering or understanding more about these pea plants as well.
Phenotype equals physical. Genotype is genetic.
Before we get started, we talk about Punnett squares in the latter of this section, but you're going to be expected to know the difference between the term genotype, which is our genetic makeup, and phenotype, which is our physical makeup. The way that I remember it is genotype begins with a gen, and it helps me remember genotype or genetic, which also starts off with gen. And phenotype starts off with the same lettering PH, which is the same for physical. So it's your physical attributes. Now, when we're talking about genotype, we're talking about, once again, that genetic makeup. And the genetic makeup basically allows us to physically appear a certain way. And that leads to our phenotype or physical makeup. Now, when it comes to more details, remember in Unit 1, specifically in Section 1.6, we talked about nucleic acids. We specifically talked about DNA, which makes up our genes. Now, this sort of touches upon DNA and RNA, which also is further covered in 6.1, which the video is already up. So if you want to, make sure to check these videos out as well. I understand that this is part of Unit 5 and we're jumping ahead, but if you want more details on that, there's a playlist link somewhere around here where you can jump to these specific section videos to get more details about DNA if you want those details. But for now, we know that DNA makes up our genes, and genes basically make up our alleles or the lettering that represent what we physically combine together to make up our phenotype, which presents themselves in our chromosomes. So when we talk about our genotype, we can have several letterings. You could have two capital letters, which is called homozygous dominant. Homozygous meaning the same, and meaning that you have two capital letters. You could have heterozygous, hetero meaning different, so I'm going to put DIFF here, and they have one capital letter, one small letter. Then we go back to homozygous and we see the term recessive at the end. Homozygous meaning same again, represented by two lowercase letter. This represents what's called the law of dominance, which basically states when there's two alleles, so you see 1, 2, 1, 2, 1, 2, the capital letter presents itself in phenotypical features. So Another way you can say if you have two capital letters, basically the capital letters are both the same and they're going to be represented in our physical features. So say we say a capital B equals the color purple, and we say a lowercase b represents white or clear. So in this case, if we have homozygous dominant, we have two capital letters, which in our legend here, we know that capital B equals purple. So the phenotypic feature is purple. Please note that these alleles, why we have two of them, by the way, is because you receive one from one parent and another one from another parent. That's why there's two. But in this case, we have the genotype, which is the genetic makeup, which makes the two capital letters, which are homozygous, meaning same. They're dominant because they're both capital letters, and so both are presented as purple. Therefore, physically, you attain purple. Now, what happens if they're heterozygous, meaning one big B and one little b? Remember, big B means purple and little b means white. So what do you think occurs here if they're heterozygous? Well, according to the law of dominance, the dominant allele, which is represented by a capital letter, is expressed, and the lowercase letter, or the recessive allele, is going to be masked. So what we have here is we have one dominant, which is the big B, and one lower b, which is the recessive. So the recessive allele will be masked and not expressed. So in this case, the lowercase b represent white, and so it won't be expressed. So instead, when you have big B, little b, your phenotypic features are going to be representative of that big B, or purple. So that's why we have the purple here. Now, what if we have homozygous recessive? Homozygous recessive means the same, meaning two lowercase letters. And when we have two lowercase letters, which is little b, basically what that means is that we have same lettering. We don't have the big B to mask it. Therefore, the little b can be expressed because there's no expression and therefore would look or present itself as white. So this altogether presents itself as the law of dominance, which basically says is if there is a physical capital letter there, those features presents themselves and the little letter is masked. However, if you just have the little letters, then the little letters will present itself. And you can see that in the lettering here. This is going to be really important when we talk about other rules and other ways to form Punnett squares, which we're going to talk about as well.
The law of segregation. Each organism has two alleles for a gene, but passes only one to each gamete.
Let's talk about the law of segregation. The law of segregation basically states that each organism has two separate alleles per gene. So, in other words, let's take a look at these flowers here. And with Mendelian genetics, he stated that each parent, which this is the P generation standing for parent, which means these are true breeding parents because we have two separate colors, have two alleles. We're going to designate this as capital A, capital A. And we know that the white is going to be little a, little a. That's what we mean by true breeding. We're assuming that they're homozygous dominant and homozygous recessive. None of these are heterozygous. And when they breed, they're going to produce all purple flowers. Why? Because dominant alleles will mask the recessive alleles, meaning that if you get one from each parent, based on the law of segregation, the one parent can only give capital A, and the other parent can only give little a. That's what we mean by true breeding parents. So during reproductions, when these alleles separate, they give one of the two from each one, right? So this parent can give only one of its big A's, and this parent can only give one of its little A's. Because it only can offer one of each, this ends up being the offspring. So each parent passes one allele to the offspring in the F1 generation, which we call the hybrid generation. So this hybrid generation ends up all being heterozygous, so big A, little a. When these cross, what happens is we end up having a 3-to-1 ratio where we have 3 purple flowers to 1 white flower. And this is considered the F2 generation. And you can see that it's 3 for every 1, 3 dominant for every recessive. And what was utilized to demonstrate this is what's called a Punnett square. So remember, we said that this generation was big A, little a, and they were going to cross with each other. So if we were going to utilize a Punnett square, which is a test cross, you draw this little box here, and let's take a look at the predictions of what this generation is going to be. So remember, the one parent is going to give big A, little a, and the other parent is also going to give big A, little a. This represents the one parent, and this represents the other parent. If we were to cross them together, we take one of each. So if sort of like a jigsaw puzzle, you take this one and you take this one. So this one would be big A, big A. You take the A from here and the little a from here, so that's going to be little a. Then on the second row, we have big A from this parent here, and then this little parent, B is the little a. Then we have the 2 little a's represented in this box. So this gives the probability of all the offspring. So phenotypically, we know that's going to be different than genotypically. Let's count the genotypes here. I'm going to use a little different color here. We have 1 big A, big A. I don't see any more of those in the offspring. 2, if you see 2 heterozygous here, 2 big A, little a, and then we have 1 little a, little a. So genotypically, this is represented as 1 big A, 2 big A, little a, 1 a, little a, 1 little a, little a. So this is our offspring that is found in this F2 generation. What this also means when we are taking a look at the physical features rather than the genotype, knowing that the dominant alleles will mask the recessive ones, we know that these are going to combine together phenotypically, meaning that this is going to be purple because you have big A there. This is going to be purple as well. The only one that's going to be white is the 2 little a's. So that's where we get to 3-to-1 phenotypic ratio, because this represents the big A, and then these two represent the big A, little a, and then this one would be little a, little a. Basically, what happens is the recessive traits appear only if both alleles are recessive, and that only occurs with the little a, which is the reason why we only see white once. This means phenotypically we have 3 purple for every 1 white, and that's what the law of segregation indicates in this section here.
This is the law of independent assortment.
So when we spoke about Mendel's 3 law of inheritance, we talked about the law of dominance and how a capital letter can be expressed when sat next to a recessive letter or allele. So if we had a heterozygous combination, according to the law of dominance, this takes precedence and therefore is expressed. We talked about also the law of segregation, that if this is a parent, each organism has 2 pairs, or a pair, I should say, so 2 letterings or 2 alleles per gene, but they only pass one of the 2 letters onto their offspring. And that was considered the law of segregation. Now we're going to focus in on the law of independent assortment. If you remember, in the law of segregation, by the way, we talked about a monohybrid cross. So that is when we had a single lettering and we tried to do a cross with the 2 alleles per trait. So if you remember, this would be one parent and this would be another parent. So this would be big A, big A, little a, big A, and big A, little a. So basically, what we had said is there are two alleles per parent. So this would be parent one, this would be parent two. And then we did a monohybrid cross, which is what this was. Now, with the law of independent assortment, we have a dihybrid cross. A dihybrid cross, usually, if you see the word dihybrid, if you think of a hybrid car, it is sort of a mix between a gas and electrical vehicle. The same holds true with the idea of dihybrid crosses, where you have two separate pairs of allele. And because you have two pairs, it's considered a dihybrid cross. Now, if you are noticing this cross right here, this is the generic dihybrid cross that's shown, and usually it consists of a monozygous dominant. So you see two pair lettering here, and there is a two sets of dominant. The capital capital letter designates yellow. So if we take a look at this, we could say that yellow is a big Y. So that's yellow. And a lowercase y, which I'm going to do a little swirly there, is equivalent to green. So this is one trait, which is color. And then the other trait we're talking about is whether or not it's smooth or round versus wrinkled. So if it was round, you have capital letter R, so for round. And then wrinkled would be lowercase, so wrinkle. And so that is another trait. So we have these two traits. And what happens is you take the homozygous dominant, which is the top that you see here, big Y, big Y, big R, with the homozygous recessive, which is little r, little r, little r, little r. So technically we call these purebreds because there's no heterozygous involved. And this is considered the parent generation. And so when we cross the parent generation, all the offspring are going to get one of each lettering because the only thing this parent can offer is big Y. So you see the big Y there. The only Y that can be offered by this parent is a little y. So you see the little y there. This parent can only offer big R. So you see the big R there and the little r from that parent there. So that is the F1 generation. So this is the F1 generation. Now, let's say that we cross the F1 generation, which is heterozygous. That would be interesting to watch. And that's where you have a dihybrid cross. So, how we designate is it's going to be a 4x4 for a dihybrid cross. So, you can see 1, 2, 3, 4. 1, 2, 3, 4. So 4 times 4 is 16. So you'll essentially see 16 blocks. And each block has to be set up with a pair. And you might be wondering where these come from. So this is going to be the parent, first parent. So I'm going to call that P1 of that F1 generation. And this is going to be called P2. That's the second parent. So this is the first parent, this is the second generation, or the second parent. Because I'm talking about the second generation, I'm just going to call this F2, this because this is F2 generation 1. That's the first parent. And then I'm going to rewrite this as F2 because this is the second generation, but this is parent 2. Now, if we were supposed to try to figure out what the lettering is, you have to find all the possible combinations. So I'm going to rewrite all the possible combinations here. The way that we use it is by the foiling mechanism. So you take the first with the first. So that's big Y, big R. That's where we get this big Y, big R. Then you take the first with the second. So first with the second. So that's big Y, little r. That's where we get this one there. Then I'm going to go ahead and take the second with the first, so little y, big R, little y, big R. And then we take the second with the first, so little y, little r, little y, little r. Because the cross is supposed to be big Y, big R, little r as one parent, and then the second is going to have the same combination. The F2 one parent is going to have the same exact, so you could see big Y, big R right here, little y, little r here. So little y, little r matches up here. Then we have big Y, big Y, little r, matching up with that one over there, and little r, little y, little r. So little y, little r. When we do the dihybrid cross, we have to go ahead and basically combo all the possibilities that you see. So big Y, big Y, big Y, big Y, big R, big R, big R, big R. So we have that one. And then you go down and combine them both. So we have big Y, big R. So that's where you see the big Y, big R. And then little y, little r, or little y, big R, excuse me. Little y, big R. So you could see all the possible combinations as you go down this chart. And if you want to manually check, you can, like to the point that you get little y, little r, and little y, little r. Whoop, that combination is going to give you two little y, little r's. And you could do the test cross and see all the possible combinations. I have all the results here. And you'll see all the potential genotypes that you can get here. Now, when you're taking a look at the genotypes of the phenotypes, this is where the difference is going to be. This is where the phenotypes of the F2 generation versus uh, the F1 generation is going to be different. So what happens is if you have all the combinations of the phenotype, they're going to be a 9 to 3 to 3 to 1. And if you take a look at how we got there, it's because we have to take a look at which ones are going to dominate on each one. So let's take a look at first phenotypically, just taking a look at the capital letters that are here. And you could see that I drew out the phenotypes here. You could see the yellows and the rounds that sort of capitalize on that. So that's 4, 5, 6, 7, 8, and then there's 9 here. So that's where we get the 9 from. Now you can see the wrinkled here, but they're yellow still. These are the 3 that comes there. And then if we take a look at the 3 green, actually there's 4 green, we see 1 is wrinkled. So 1 is wrinkled. And then the rest is going to be smooth. This gives the generic ratio of 9 to 3 to 3 to 1. I would actually commit this to memory that if you have a test cross with a heterozygous of 2 traits on a dihybrid cross, that it's always going to come to 9 to 3 to 3 to 1. Just as a hard and fast rule, this tends to come up a lot in terms of problems that you understand the phenotype that's associated with the dihybrid cross, specifically with the traits of heterozygous times heterozygous. Now, genotypically, let's look at the difference here with these numbers, which is going to be a little bit more complicated. We have 1 big Y, big R. So we have 1 big Y, big Y, big R, big R. And you could see that's the only one that exists. So I'm just going to go ahead and cross that out because we talked about that. Then we have big Y, big R, little r. Now that only comes in twice. So big Y, big R, little r, big Y, little r, big R, little r. So we have 2 big Y, oop, big Y, then big r, little r. So you could go ahead and try to see all the possible combinations, and you'll find that there are a ton of combinations. There's 9 different genotypes specifically, and it goes like 1 to 2 to 1 to 2. And you don't need to know that specifically. You would just do the dihybrid cross and check. I would not commit that to memory. That's not usually what is tested in terms of like specifically dihybrid crosses, but this is a hard and fast rule. If I was given a dihybrid cross, I would actually do the whole test cross if it came back down to memorizing the genotype and getting all the combinations. You are given multiple choice questions, so if given a multiple choice question, they would most likely like be which one by multiple choice, and you use proper process of elimination to see which ones are there. So, but this just shows you how to do the cross for now. And the key unique things to note is that one, we do have the 9 to 3 to 3 to 1, and that the ugly ratio of the genotype, do not memorize it. And the phenotype you should know for that dihybrid cross if you have 2 heterozygous. And what does this tell us in terms of law of independent assortment? If you remember in meiosis, and we covered meiosis previously in this unit, so if you remember, we covered meiosis and mitosis. And in meiosis, there are 2 separate parts. I would suggest going over video 5.1 if you want more of a detail, but there is a step metaphase 1 where homologous chromosomes line up at the metaphase place. To recap, homologous chromosomes are a pair of chromosomes. You receive one from mom and one from dad. And if you remember, these help assist, meiosis specifically helps assist in creating gametes, which creates the egg and sperm used for offspring. But in this case, what happens in metaphase, the homologous chromosomes will line up at the equator at metaphase 1, and they line up randomly. So you have no idea what combination you're going to get. There is no rule that says, okay, the paternal one is on the left side and the maternal one is going to be on the right side. No, you could have a combination where literally it could be the most random color of chromosomes that match up. So if one chromosome carries, for example, the gene for seed shape and another one carries seed color, the orientation of these chromosomes are independent of one another. So that's what the law of independent assortment says, that the alleles for various genes separate independently in that step of meiosis, and because of this, one trait allele does not affect another trait's allele. So when we're doing these dihybrid crosses, we know that these genes are on different chromosomes and they're far apart. So this is why we are basically trying to predict all the possible ratios and probability that we can have. The chromosomes line up randomly and line up at that metaphase plate randomly at my metaphase 1, so we don't know what our combination is going to be, so it's completely random. And you can see when you're doing heterozygous all the possible genotypes you can get and all the possible phenotypes you can get. And this shows us how genetic diversity is so important. The randomness is what allows for genetic diversity, because this means that we lead to gametes that are going to be different. And so we know that we need meiosis to be random to allow for that genetic diversity.
By studying pea plants, I discovered that traits are inherited independently, revealing the rules that govern inheritance.
Let's talk about how fertilization affects inheritance. When we talk about fertilization, we talk about this idea that there are 23 chromosomes in each haploid for humans. When the egg and the sperm join together, which each have 23, one from maternal and one from paternal, they join together to mix alleles, therefore combining to eventually make 46 chromosomes, which is characterized as diploid. This creates a whole new allele combination and increases the genetic diversity. When we talk about this, we note that we spoke about the law of segregation and law of independent assortment, and how homologous means same chromosomes and heterozygous means different chromosomes. Now, if we talk about patterns of inheritance, we notice that there are autosomal inheritance, which means that if I go ahead and say big R, little r, let's say, for example, the big R is characterized as having a disease. So in this case, if we have autosomal dominant, this means that we are affecting 22 potential chromosomes, because remember, the 23rd is considered XY chromosome based off the sex chromosome. So because of this, we'd say the 22 are going to be considered autosomal. And so if it's autosomal dominant, that means that the capital letter is what dictates the disease. So let's say we have an affected father, that's going to be big R, and we don't know if there is a little r involved just yet. But if there's an unaffected mother, remember that just means that she's recessive because she can't have the dominant gene. Now, that's to also note that if she did have the dominant gene, she would have the disease, but in this case, she doesn't. And you can see there's unaffected children, which means that they had two little r's as well. So because we have the unaffected children here, we can note that one of the parents, the one that was affected, is going to be heterozygous because they had to give their little r to their offspring. And the affected children are just going to be big R, little r. We know this because the mother can only give the little r, and big R, little r for the children. So this is an example of autosomal dominant inheritance because it shows you how the alleles play into a part of the offspring and the chance of having a disease. Now, if you go ahead and take a look at X-linked, this means that you can have a chromosome issue or a sex-linked related gene. So the gene is located on the X chromosome. And for example, if you have a disease that is recessive, let's say we have designated big L and little l, but we know that it's sex-linked and it's on the X chromosome. So for example, let's say that we have a carrier mother. They have two X chromosomes. So if they're a carrier for the illness, that probably means that they have big L, little l, but it's attached to the X chromosome. Now, an affected father means that they have the recessive allele, the little l, specifically on the X chromosome. So what happens in these sex-linked, X-linked related diseases, because the males only have a chance of it attaching to the X chromosome, they tend to persist more or be more prevalent. And you could see that in this case because either you have the disease or you don't have the disease. And then what ends up happening for the offspring is for the males and females, if they are affected and it's recessive, they're going to have the little l attached to the X chromosome, the sex-linked chromosome. If it's affected, then basically what happened is the carrier female's little l and the male's X that's attached to the L, so two little L's attached to the X, allow this female to be affected. So this just shows how sex-linked chromosomes, which basically means that they're attached to this X chromosome, play a part into the probability of having the sex-linked inheritance. Now we can look at linked genes. Linked genes just means that you can have a gene on a chromosome, usually with another gene on the chromosome. So let's say there's two of these on the same, they're passed together. So when they have crossing over, the location of these are passed together as to whether or not you have the disease or you don't have the disease. And so that's very important to note. When we take a look at pedigrees, we take a look at the understanding of what this looks like. So first of all, they're oftentimes shown with several generations. So pedigrees are visual images to basically diagram inheritance across multiple generations, and they help identify basically dominant versus recessive traits, and they can infer potential genotypes, especially when it comes to autosomal and sex-linked inheritance. So each generation is its own line. So this would be the parent generation, this would be the first generation of that parent, and then this would be the second generation. So you would see these are the little indicators there. So this would be considered a man because it's a square, and this would be a female. So anything that's considered a square is a male, circle is female. When they join together, that's going to be the combination and potential offspring that they have. And so this is an offspring, this is an offspring, and this is an offspring, and this is an offspring as well, because they come from that arrow. When they marry into it, you could see here, this is married into it. There's no connection coming from here to the parent. So you could tell that's someone outside coming in to potentially mate for the following generation. So we know that squares mean males and circles mean females. Other things to note is if they're darkened, that means that they're affected with something. Usually a dark color indicates if they're affected. Sometimes you'll see a mix between the two. You could see some of these are affected and then there's nothing in there, and that usually means that they're a carrier. So what we mean in terms of alleles is if we had a carrier, we would say, we said that they're a sex-linked carrier. Remember, XY, this would not be an X sex-linked carrier because they would have to have the disease. But let's say in this case, it's a disease that is considered to be linked. Then you would say, for example, heterozygous, the lettering big L, little l, would designate these individuals, the ones that are mixed. Those that have the disease are two little l's, and the ones that are plain circles like this one would be big L, and then we don't know about the other one until we take a look to see if they're a carrier or not. So for example, the affected one has little l, right? So we can tell from the little l that these two are little l for the female. And if they gave one little l, this means that this unaffected individual had to be also one to give. So this is just an example. This is not a perfect test cross. It's just an example to show what a pedigree would look like if you were looking at the different symbolism. And when you take a look at it even further, you can even distinguish other characteristics as well.
By tracing family lineages, pedigrees allow us to track alleles across generations and predict their inheritance into the future.
Let's talk about this idea of autosomal dominant versus autosomal recessive. First, let's take a look at autosomal recessive. The white color here represents not having the disease. So if something is autosomal dominant, that means for this side, this would be two little r's as not having the disease. Whereas if you have autosomal recessive, that means that they have the disease. So that would be two r's here. Which means that if you don't have the disease for autosomal recessive, you're going to have a big capital R in all of these situations. In this case, because the individual has the disease, they're going to have to be heterozygous to pass on the illness onto this one offspring to be autosomal recessive. Oftentimes it skips generations, but if two unaffected parents in the opposite way were to have the affected child, the trait can't be dominant. Both parents have to be heterozygous for the offspring to have the disease. Now, if we're talking about autosomal dominant, basically that means those who have the disease are going to have capital R in the disease, which basically means that if two parents are affected, the chances that they're going to appear in every generation. The trait can't be recessive. The parents have to be heterozygous for one individual to come out as not having the disease. So we could see the difference between these two pedigrees. Of course, the pedigrees would be more expansive, but this is just giving an example in short form. Now, if I had a pedigree that was X-linked, you could have X-linked dominant versus X-linked recessive. And just quickly glancing at this, if it's X-linked dominant, you'll find out that if you have an affected dad, because if you have X-linked dominant and you are a male, that means that you have the combination of XY and you basically have a big R. All your X chromosomes can only be offspring that potentially give it to females who have two X chromosomes that big R. So the affected dad always gives it to their daughters because their only X chromosome is going to be passed along. In opposite, if it's X-linked recessive, if you have an affected mother who has X chromosomes, which basically means that they are 2R lowercase letter, the sons, which have an XY chromosome, basically can only receive an X from their mother. And if they receive that X from their mother, because the father has to give the Y, that X chromosome will have to be the one that's affected. So you'll notice that the pedigrees, if it's X-linked dominant, have a whole generation of females that have the disease. And then if it's X-linked recessive, then all the males have the disease. And so that's how you could tell the difference. Now let's try a test question. Pause the video and then unpause once completed. Okay, so let's take a look at this to create the pedigree and the answer. First, let's check to see if you got the correct answer. The correct answer would be 25%. Let's see how we got there with our test crossed. Firstly, what we're going to do is we're going to do dihybrid cross. So we have the first with the first. So that's going to be big B, little l, so that's what we have here with the one parent. Big B, little l again, so we have that again. Little b, little l, and little b, little l, which we have on this side. Now, if we take the other side, we have the little b, big L, little I. So for this one, we have little b, big L, which is what we have here for the second parent. Little b, little l, which is what we have for that second parent. Then we have little b, big L. Once again, it's there. And little b, little l, which we have there as well. Basically, we have to find all the combinations and we'll find it here. It says which one is going to have brown body and short antenna. So brown body is going to be recessive, which means that we're looking for two little b's, because if anything has a big B attached to it, it's going to mask it. It says short antennas, which once again is two little l's. So we're looking for homozygous recessive. We only see it 4 times out of the 16. And if we were to reduce this fraction, it'd be 1/4 or 25%. Therefore, our answer choice is B. Hopefully this helps you answer pedigree questions as is.
Appreciate you riding along for our cosmic journey. If today's mission brought you the knowledge that slaps, make sure to hit that like button to boost that signal. Subscribe if you're locked in for our next launch. And don't miss the entry. It's got all the goods for AP Bio, PSAT Math, and SAT Math. Before I sign out, make sure you're all in one orbit and diving in. Killing out. Till next time.